Okay, today we're going to be talking about altered gene expression due to predator stress in both individual Pardosa milvina and in their offspring. So, the environment has a wide range of effects on many animal species, including helping to shape the expressed phenotypes of many animals. Phenotypes, of course, are rarely as simple as just one gene producing one phenotype, and in many cases, the environment does play a role in how the genes get expressed. One of the ways that the environment can alter the way that phenotypes are expressed is through um, gene expression levels. So essentially, the number of RNA copies of a gene that is getting made, and affecting the number of proteins produced and how strongly or how weakly that gene is being expressed in the individual. Now, there is growing evidence that this is not held to a single generation, but you can have transgenerational effects as well. And for those who are not familiar, transgenerational effects here, or transgenerational plasticity, are, are phenotypic or behavioral changes in offspring induced by the environment of the parents. So we have a little cartoon here. We have a dog. Dog has a nice standard black nose in the parent. Something environmental stress occurs, and you see in the offspring, we see a phenotypic change. The nose is now white, and we see that persist into the F2 or grand offspring. So this is the star of today's show. This is Pardosa milvina. This is a small generalist wolf spider. They live in agricultural fields. They are interesting as they have a very stereotyped behavioral response to the much larger wolf spider, Pardosa helio. Larger wolf spider naturally preys upon Pardosa milvina. And we see things like reduced movement, reduced feeding, and heightened vigilance when exposed to chemical cues from Pardosa helio. And we also see that the Pardosa milvina have transgenerational effects due to these chemical cues from predators. So what did we do? Well, we exposed a number of Pardosa milvina to chemical cues from this guy, Pardosa helio. You can see him actually eating a Pardosa milvina in this picture. And we went out and we collected Pardosa milvina. We split them into two groups, a control and a predator exposure group. Um, Predator exposure group was exposed to chemical cues from Pardosa helio, largely silk and excreta. So we would collect these cues on filter paper. The predator exposure group was placed on filter paper with chemical cues for 1 hour twice a week, and control spiders were placed on blank filter paper. We also allowed the both females of both groups to reproduce and to mate and produce offspring who were never exposed to predators or predator cues themselves. So the first hypothesis we tested here was that exposure to predator cues would cause gene expression changes in Pardosa milvina. Pretty straightforward. We see behavioral changes, we might expect some gene expression changes. Second hypothesis we tested was that maternal exposure to predators would cause gene expression changes in the offspring of these predator-exposed spiders. So what did we do? Well, we took 6 predator-exposed and 6 unexposed adult females. Now, all these 12 females were virgins. We had not mated them. And then we took 6 third instar spiderlings, so small, still very young spiderlings, from exposed mothers and 6 from unexposed mothers. All the males used for matings were predator naive. We extracted total whole body mRNA from these spiders, and then we constructed 3 libraries per group, so a total of 12 libraries from those spiders who we extracted mRNA from. Using the 3 per treatment group, they gave us the highest level of mRNAs. These libraries were then sequenced by Novogene, which is a company that does all kinds of sequencing for genetic analysis. And we compared the level of gene expression between exposed and unexposed adults and the third instar spiderlings. So what did we find? So first, if we look at the level of gene expression we see in the adults, these are the ones who are actually exposed to spiders. Anything in red on this graph is upregulated. So you see more copies of the mRNA in spiders who were exposed as opposed to spiders who were unexposed. Anything in green in this graph is just the opposite, it is downregulated. So fewer mRNA copies in exposed females versus unexposed females. Anything in blue is not differentially expressed. We had 133 upregulated genes and 776 downregulated genes. On the flip side, look at the same kind of graph, but for the juveniles. So these spiders were never exposed. Their mothers were either exposed or not exposed to predators. Same concept. Anything in red is upregulated. So offspring of exposed mothers overexpress these, and anything in green is downregulated. Offspring of exposed mothers underexpress these. Anything in blue not differentially expressed. Here we saw 178 upregulated genes and 111 downregulated genes. So the next question is, well, what do these genes do? What are the function of these genes? And we looked at that as well, and we'll start by looking at upregulated genes. We see that in both adults and juveniles, venom or toxin genes were upregulated. These are almost certainly venom production genes. We saw metabolic rate or metabolic activity genes being upregulated. So this makes some sense. If the spiders are being stressed, that is certainly going to affect their metabolic activity. In adults, we saw response to chemical stimuli genes being upregulated. This is exciting. We did expose these spiders directly to chemical stimuli, so they are showing a heightened response to the kind of stimuli that they received. In the offspring, we see drug binding genes were upregulated. This certainly could be exciting as well because these could be functionally chemical binding sites. We also see stress genes being upregulated in at least in the offspring. So certainly, predator exposure could be stressful. As far as downregulated genes, in both groups we see DNA repair genes being downregulated. And we see zinc finger protein genes being downregulated, many more in the adults than in the juveniles, but some in the juveniles as well. This is exciting because zinc finger proteins, among many other things, act to help regulate gene expression themselves. So these genes being differentially expressed could be affecting a lot of the other differentially expressed genes that we see. We also saw things like apoptosis and heat shock genes being downregulated, particularly the offspring. We saw a few molecular pathways that were differentially expressed, particularly a vasopressin regulated water reabsorption pathway was upregulated in adults, and we saw an alcoholism and a transcriptional misregulation pathway being upregulated in the juveniles. So I'd like to thank a whole bunch of people, of course, my committee, Miami University, for all the support, all the undergrads who work in the lab and make doing things like research possible, a bunch of grad students, particularly Patrick Garrett, for help with a lot of this genetics work, the American Vaccinological Society for funding, Doctors Andrew Kiss and Rich Moore for help with assistance with all this genetic stuff, and of course the Animal Behavior Society for allowing me to present this research.